So before we get into the interview, how about a little bit of stem cells 101? So why are stem cells so special? So we've got a stem cell and we get a magician, and no, we don't get a magician, we get a scientist to do some magic in the lab, and what they can do is they can make that same stem cell, replicate it. They can make it perfectly identical. So that's really amazing. That's the potential of that stem cell. And also, a stem cell can go back to the lab and then be changed into another type of cell, which is called specialization. So that's what makes them so special.
So reproductive cloning is where you're trying to make a copy, a clone of that original donor, whereas therapeutic cloning, what you're trying to do is make their stem cells. Now, that whole area, of course, was very exciting when Dolly was born, because it showed that the technique worked. And we've gone on now to show that you can make stem cells, even from human cells. But I think it's worth mentioning that the technology has also changed in those 10 years. So the technique that I was very much involved in developing, and I worked in mouse, but I was very interested in whether we could apply this in humans. It's still important, but there's a new technology called Induced pluripotent cell technology that in some way kind of supersedes it. And I think it's a great example of how quickly science evolves.
Did Megan say induced pluripotent stem cells? What are they? That sounds like a really big concept. Well, not really. So don't Google it, just listen. For a while now, we have been obtaining stem cells from human embryos. So very clever scientists are able to utilize the stem cells from a human embryo and transform these into different cells with different characteristics, such as a muscle cell, a heart cell, or a kidney cell as an example. That means they have multiple potential. That's called pluripotent, multiple potential. And induced stem cells do not require a human egg to grow. This is the interesting science behind it. This is probably information for another video all entirely, but it's really amazing science. So you can get a skin cell, load a virus with new genetic information to create a different cell, a heart cell or a kidney cell or a muscle cell.
To some extent, well, yes, the simple answer is yes, there are risks. And some of the risks are the unknown. So in a lot of science, we don't know what happens when you add A plus B. You want to observe the process, you want to see what happens. And with stem cell biology, we know you can do certain things. We've worked out how you can mimic early development. So we've worked out how you can mimic early development. So we can take a stem cell from lots of different sources, give the cell the right signals, and it will turn from a stem cell into a nerve, or from a stem cell into a kidney cell, or from a stem cell into a heart cell, a muscle cell. So we can manipulate all that. What we can't do is do it absolutely, and we can't kind of dial it up. So it's not at the level of automation where you can just kind of key in a sequence and get an absolute product, but we're getting closer. And so in the 15 years that I've been involved in this space, I've seen this huge evolution in terms of not only what cell types we're using, what stem cell types we're starting with, but how we do that manipulation, how we kind of coerce and push the cell towards the stem cell, how we push the stem cell to a particular fate. So that's all really exciting. It's new. In terms of risk, I think the other point to ask, or the point, so maybe it's less of a risk and more of a question that this technology raises, and that is, what is that cell? So we know we can start at a stem cell, we can make it into something that looks like a nerve, but does it function as a nerve? And when it comes to starting to apply these clinically, how do we get them to the patient? So it's one thing to kind of grow a nerve in a dish, but how do you transfer a functional nerve or a nerve into the body that's going to be functional, will integrate, and will help restore the loss of function in a patient? So for me, the knowledge allows all these possibilities, but doesn't yet provide all of the answers. And I think that's what keeps the field so alive and so interesting.